Welcome to the developmental biology. Here you can see 3 different videos. In each one of those, we're showing 3 different model
The
organisms.
fertilized ovum
Here you
undergoes
can
rapid
see
divisions and forms a
this
blastula.
one frog,
Each division
here
normally
is
takes about 30
zebrafish, and
minutes.
here are fruit flies. And in each one of those, you will see that in early development, there is a lot of different processes that are taking place. And during this course, we're going to analyze what are the signals, what are the cell behaviors, and what is happening with the tissues to produce all this diversity of movements and all the diversity
blastula
of
develops
shapes.
into a
And also
gastrula.
what happened during evolution to change those things. Okay, I hope that you are excited to learn more about developmental biology, and let's start. Okay, this class will be divided in the 3 main sections. In the beginning, we're going to have some questions about developmental biology. Then we will talk about history, and we are going to focus on this debate between preformation and epigenesis that it was in the beginning of developmental biology. And later, we will talk about some of the presentations that you will give in this class at the end of this class. So let's start with some of the questions that developmental biology has. Okay, I want that you think about this. Is this true? Photoshop, AI? This could be real, this cannot be real. And why do you think that why somebody is going to grow an ear? Yeah, the cartilage, of an ear, the outer part, on top of a on top of a mouse. Why do you think? If this is real, and what is the purpose of that? And if it's not real, is it will be useful or not? And the answer is yes, this is real. Here, if you see here, do you see? And we zoom in here, you can see that in humans, they can do something similar. And what they are doing is growing tissues that on top of tissues, and they are just, as we know, the signals and know with the mechanical pressure and all the molecular basis, we can grow tissues. And for example, somebody that lost their ear, they can grow it in their arms, then remove it and put it in the ear. So this was actually true, and this is something that we are getting better and better in doing that. Okay, now let's watch this video. And again, is this video true or is again AI? So here what they are doing is using a spinach leaf for repairing hard tissue. Yeah, so they bleach the leaf and they keep all the structure, all these veins that and look, you can see how they have all these small tubules. So true or false? What do you think? So the answer of the Spanish was that that one was also true. Yeah. So I'll show you two things in developmental biology that are very important. That is that we can use it to understand better the principles that can help us to cure diseases. Yeah. But not only that. So they can also help us to understand better uh, other principles of life. Yeah. So and this one is fact, true, belief, opinion, and alternative facts. And during this course, for example, I'm going to tell you things that to give you more tools. Yeah. So for example, to take a better decision. So for example, this was is a very conservative politician, and yeah, this and he said that pro-life is scientific. Biologically, life begins at conception. That is irrefutable from a biological standpoint. Um, so he said something. A politician said that scientists consider that this is true. And if you talk about that with a scientist, we are very confused about when we say that life starts. Yeah, if life starts when the nervous system appear, yeah, because a cell is alive, but do we have to protect any of the cells of your skin? Yeah. Or do you think that is when the embryo is formed? Yeah. Or it should be before that? Yeah. So it's very important to study developmental biology to have better tools to understand, to take a position in very important discussions that are affecting our uh, democracy and the future and the decisions that we take. There are articles that even say that conservative and liberal brains, they might have some difference, and you start developing into your brain, start focusing on something different when you're a liberal and when you're a conservative. And we have to study that in more detail too. And that is part of developmental biology. Finally, one question that I have in my as a researcher is cancer ubiquitous disease? Yeah, I'm very interested in understanding cancer. And is cancer located in multicellular organisms? Some plants can get cancer, or is only located in animals? And what other factors affect that cancer appear? So, for example, there are animals that don't get cancer or get very little. So sharks get very little, mole rats, and we don't know the principles. And let's talk about some principles that developmental biology can help us to understand better. So let's think together about this question. So, for example, do you think that the diet is important or not? So animals that are carnivorous versus herbivores or others, do you think that the diet is important? What you eat will determine that you get cancer or faster than what, yeah. Carnivorous, if you eat meat constantly, you will be more prone to cancer. And the answer is, according to studies, yes. Animals that are carnivores are more prone to cancer than the herbivores. But about in the phylogeny, and in the phylogeny there are organisms that we don't know well why that are more prone to cancer, and other ones can live for a long time. So we're going to talk about the age, animals that seems that they don't age in the same way. And what about the size? Yeah, yeah. If you have a big whale is not more likely that the big whale, as he has so many cells, it will have more cancer or not. What do you think? So look, the lifespan is here. Here's the whale that lives around. This one lives around 30 years, so between 30 and 40. Here you have humans. Here, man, is here. And look, the turtles. So why some animals live longer than other ones? We're going to talk that during in developmental biology, and in humans, definitely something associated with age is cancer. As we start accumulating mutations, now we know we're going to talk. If you take cancer biology, you will see that there is a strong correlation between, for example, the smoking and 20 years later developing cancer. So this is something that is fascinating, and in developmental biology, we study these kind of questions. And now I'm going to add a video that I found fascinating about the large size and the development of cancer, yeah, that they are not correlated. I cannot attach it to this video, if not, they will not allow me to upload it in YouTube, so I put it later so you can watch it, but it's fascinating. Okay, so to finish this introduction, I show you a very crazy picture that you might think that they are false or they're Photoshop or at least there are gross, but actually they are very interesting findings and very interesting projects in research that we will see if will become a reality in 5, 10, 20 years. And I also show you some questions, yeah, about cancer that and mortality and size and all these things. If you're interested about the question about big animals and why don't big animals get more cancer, here is the explanation. So you can go and give it a look. Um, and yeah, I just wanted to send you a lot of questions and show you that developmental biology has many of these questions that is possible to answer and that is looking to answer. Okay, so with that, I just want to motivate you to get excited into this field. Okay, so we just finished the section about the questions, and I show you some of cool questions about cancer, in particular, uh, sorry, some questions about developmental biology, and I focus on cancer because this is the area that I work. And rather than telling you this is the answer, it's like this is the hypothesis that we have, and maybe in the future we will find the answers. Now we're going to talk about the preformation and the epigenesis to start talking about the history of developmental biology. This section you will see it in a lot of more detail in this course and this link that I cannot put it in the class in this video because I cannot upload it to YouTube, but I will put on the link there so you can keep watching it too, so you listen it in more detail. So very briefly, I don't know if you remember Aristotle. Yeah, if you took genetic revolution, I'm sure that you remember him. And so he was one of the first person that have this idea of preformation of epigenesis, and and we studied this in the in genetic revolution, and also I don't know if you remember this preformation and epigenesis and this homunculus idea that there was a tiny human being inside of the sperm and or a tiny woman there, yeah, and inside of them there was even a tinier human, and inside of them there was a tiny human. So it was that idea of the Russian doll. So now we know that this preformation idea that the body was preformed is not correct, but there is a process of epigenesis that we start from molecules that they need to reorganize. Yeah, and this is a very important story, part of the story of the development of this field. That if you watch the video, you will listen to that in more detail. So, but to summarize, and to remind you what we talk about in developmental biology, Aristotle considered two basic developmental questions. Yeah. So here he considered this hypothesis that there was only growth, yeah, so the animals, and he used eggs, yeah, embryos of eggs, and so what he found is that the first hypothesis is that the preformation idea that the eggs they are just inside of the eggs in the early beginning there was just very tiny chicks, yeah, that start growing and becoming larger and larger, yeah. Or the other idea it was this, that there was a stepwise process that show a progressive organization, and that later from that process there was an increase in complexity that led to the development of the chicken, yeah. So in other words, I will read this question. The first question it was, do all parts of developing organism come into existence together or simply grow larger? Yeah, so this is the first example, things just grow bigger. Or the other one is that there is a stepwise process and there is a process of organization. And what we found is that what he found is this one, yeah. So the idea of the epigenesis, right, that the preformation is correct, yeah. And the homunculus idea is definitely incorrect. And here I add, so if you check in a short term, yeah, you can think that maybe this is only what is happening, but if the earlier that you go, the more clear is that there are different changes in the eggs, and that the correct answer will be this one, the stepwise process. And his observations support the concept of epigenesis, uh, but this term was not used until mid late of the 17th century. Okay, yeah. So for many years, and if you remember, uh, in the past, the first microscopes didn't have a lot of resolution, as we covered in genetic revolution. So in the beginning, actually we thought that the preformation was the correct one, and we forget about Aristotle. But then the idea of epigenesis came back. Okay, so and now, although I wanted to make it as clear as possible that the preformism was wrong and the epigenesis is right, I'm going to give a little bit of jump that always happen in science. That is that sometimes something new methodology appear and it changed things, and the microscope did that. However, in this case, it went a little bit backwards, and the microscope didn't have enough resolution, as I said before. So some ideas came back, and the preformism went back for a little bit. Yeah. So let's talk about that. So between the 16th century and the 18th century, this debate came back, and here you have the preformation, where the entire human is inside, and the epigenesis, that the body structures were not present in the sex cells, but they're formed anew. Yeah, that's exactly what happened with the example of the chick. Yeah, that in the embryo of the chicks, everything is disorganized, and over time they form an entire chicken. Okay, so here you see a beautiful diagram from somebody that at that time with a microscope started drawing, and if you see with the resolution of the microscope, everybody would thought that this could be what is inside there, yeah. And here there's a beautiful diagram, but although it's beautiful and it's in a lot of detail, it's not at the cellular level. So it could be very difficult. And I want that you think a little about this question. Imagine this. How do you think that structures appear in the course of development from structureless material? Yeah. So that question, we still have that question. So at that time, it was much easier even to think about the preformism about growing, yeah, than about epigenesis, because epigenesis is the most difficult answer. That and this is why maybe it was easier to take the side of of preformation. So for the preformist, the sex cells contain the complete miniature adult, or the homunculus, that this is how they call it. And I told you that, and I put as an example this Russian doll, that this is a theory, yeah, some radical ideas that you see that the Russian doll have one smaller one and one that is smaller one that is smaller. So this is what they thought about the 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 sex cells. That inside of that there will be a tiny human, and in the sperm of this tiny human there will be another one, and so forth, even back from Adam and Eve. Yeah, so there was a combination between religion and science, and thanks to the development of microscope, we were able to see now that inside of the sperm what we found is a nucleus and organelles, cellular organelles. So there was known that it was a hypothesis that it was discarded, and that the more closer to the reality is the epigenesis. And if you think, the idea of development at that time is already exist, but it was only in terms of growth. As with the picture of the chicken that it slowly start growing and growing and growing. Here, this is what they have. An idea of growth, but no epigenesis. Now we know that is the combination of both. Growth is important, but in the early beginning, we need the idea of epigenesis. And here there is a lot of names between important preformists. Yeah, great scientists, and here epigenetists. Yeah, you don't have to remember all the names. What I want to tell you is what we talk about in Cells and Energy, what we talk about in all the revolutions, the courses, is that the key here was to develop experiments. Yeah, so to go develop a very important techniques that allow us to have better microscopy images, to know that the early microscopes, yeah, they were producing false positive due to the resolution of the microscopes, and create some people that create that it was in the sperm, or ones create that it was in the oves, in the eggs where the homunculus were there. And the problem that we had, that the epigenesis had, is that there was not a potential mechanism to understand that. Yeah, so although the epigeneticists, they were correct to some degree, yeah, there was still, and we are still finding the mechanisms. And during the class I will talk about some of the mechanisms, and I start showing you all this complexity that happened in the beginning, just to tell you, we now can image much better, and there's so much complexity, and we are going to talk about some of the things that we already know. Okay, hope that you are excited as I am excited. We are almost done with the first class. Okay, so now I want to show you a picture, not a picture, a movie of a light microscopy, similar to what the first microscopy people observed with sperm. And as you can see, it's just very difficult to observe anything. Yeah, and it's just cool to think that maybe when the sperm died, you will be able to image it better. It will be easier, but as you see, there was not enough resolution to see what is happening there. Okay, so to finish this class, I'm going to tell you, okay, so I have been telling you the preformation was incorrect, the homunculus wasn't there, and that is correct. But I want to tell you to go beyond that and said, okay, so was what we learned about that lesson? Yeah, I think what we learned is that none of them were completely correct. Yeah, neither was totally correct. Like, and there was definitely in the epigenesis, there was a gradually a stepwise process, and this is clear. Yeah, so I show you in the beginning of the class all these videos, and I show you here another model organism that show all these embryos, how they develop and form shape, and that is completely correct. But these don't start from a highly disorganized process. Yeah, actually at the cellular molecular level, what we found is not something that is very disorganized. What we found is, for example, here, this is the gene expression of the early embryo in Drosophila, and we're going to talk about all these patterns that happen in the development. So although it seems very disorganized, it seems that there is no organization, there is a lot of organization happening from the beginning, and not only at the genetic level, at the cell division level in Drosophila, and the movement of the cells. There are so many things that seems to be that are very organized even from the beginning, and we are still finding those things. So both have part that they were correct, both have part that they were incorrect, and there is a lot of work to do. Another part that is that is I want to talk, and maybe in class we will discuss more this later, is about the new preformation ideas. And one of the new preformation ideas is that all the information is in the DNA. Yeah. So now in the new homunculus could be the DNA. Why? We have all the information at the DNA. We don't need the cell to interpret all the information. There is no changes. We have the DNA, so everything is formed there. And if we take that idea to the extreme, or in the beginning when the DNA, with the Human Genome Project, we thought that we are going to be able, as we decode the genome, we were able to cure all the disease. And what we found is that we need more about the epigenesis to understand the mechanisms underneath that. That is a really good tool, but it's not enough to do that. Yeah. So understanding the DNA, yeah, could become the new homunculus, and is not enough to understand each one of the components. And do you remember that I told you that there were scientific articles that they think that there is a possibility that our brains between liberal and conservatives work differently? Same thing with preformation and epigenesis in biology. So sometimes some scientists are closer to think about, it's easier to think about epigenesis, and for other ones it's easier to think about preformation. For example, when I study biology, the ideas of docking, yeah, it was understood that the DNA was like the blueprint, and it was a very useful and powerful analogy that it was enough to understand everything in the DNA. These ideas currently have been evolving and have been becoming less strong, and now developmental biology is getting a new power to understand some of those things. Okay, today we talk about key questions on the introduction to developmental biology. Then I did a brief review on preformation and epigenesis in the past and the present. And in the next section, in the next video, you will have some guidance to talk about the presentation that we're going to give in this course. Okay, I hope that you enjoyed this class, and if you have any questions, let me know.